Hi students, today we're going to talk about the 5th chapter related to how neurons communicate with one another and how they adapt over time. This will bring us to the end of the material that we'll cover for the first midterm. So after you do this 5th quiz, the next thing on your plate will be the upcoming midterm next week, in which there'll be more questions from the chapters 1 through 5, and the information is on eClass. So today we'll be talking about 4 topics: the nature of the chemical message that's sent between one neuron and another neuron. We'll talk about the varieties of neurotransmitters, the messengers that are used to send these messages, and the receptors that receive them. We'll talk about kind of general systems of neurotransmitters and how they're related to behavior, and then we'll talk about how synapses can adapt over time and introduce this idea of the brain changing with learning. So the first experiments done to understand how neurons communicate with one another were done on an animal's heart. And it was observed that the heartbeat quickened. Oh, it's known, of course, that our heartbeat quickens if we're excited, and when we go to rest, it slows down. And so it was discovered that chemical messages were actually sending these signals, either excitatory messages to speed up the heart or inhibitory messages to slow it down, in these interesting experiments. So the heart of a frog was removed and kept alive in a saline bath, and the vagus nerve that connects to the heart was stimulated with electrical current. And a recording device recorded whether the heart was beating. And so here on the graph, you can see the rate of heartbeats of the heart. And you can see that when the vagus nerve into the heart was stimulated, the heart rate slowed down. Now, importantly, the experimenter connected the fluid cavity of the first heart to a second heart, so that the only thing connecting the two hearts together was the common fluid that they were in. And observed that when the first heart was stimulated, even though the second heart wasn't stimulated itself, the heart rate slowed down in that heart as well, indicating that whatever made that heart rate slow down was something that was dissolved in the fluid. And it was proposed that was something that was released by the neurons of the vagus nerve onto the heart muscles, and therefore dissipated into the fluid to the other heart. So Otto Loewy did these experiments in the early 1920s and showed that the neurotransmitter acetylcholine, or we write ACH, slows down heart rate. So acetylcholine was the first neurotransmitter discovered in the PNS, the central nervous system. It was observed that it activates skeletal muscles like in the somatic nervous system out in our body, and it may excite or inhibit our internal organs like the heart, depending on the connections in the autonomic nervous system. Otto Loewy went on to understand that epinephrine had the opposing effect on the heart. So stimulating a different nerve into the heart mobilized the heart and made it beat faster. And he discovered, therefore, that epinephrine had this excitatory or mobilizing effect on heart rate. And so therefore was a neurotransmitter in the central nervous system. And norepinephrine is a related neurotransmitter found in the brain and in the parasympathetic nervous system, and it accelerates heart rate in mammals. So these are 2 other types of neurotransmitters. So a neurotransmitter, more generally, is any chemical that's released by a neuron onto some other target, where on that target it has either an excitatory or inhibitory effect. And one way that neurotransmitters influence our body is by circulating in the bloodstream. And even some of the same chemicals that we consider neurotransmitters in the brain, when they're circulating in the bloodstream, we consider them hormones. So these hormones circulating in the blood have much slower action because they require our blood to flow to that area. So we'll learn in this course that the hypothalamus activates the pituitary gland, which releases hormones into the blood that targets our organs. And these are separate from these neurotransmitters, which are contained inside the brain. In order to understand the structure of a synapse, the electron microscope images of these synapses in the 1950s were needed to better understand the microscopic architecture of the synapse and how what it contained. An electron microscope projects a beam of electrons through a thin slice of tissue, and different structures in the tissue scatter the beam differently. And so the received light, the received electrons can be therefore used to recreate an image of what was the substances of that slice of tissue. And this is much better resolution than a light microscope, which you remember looking through in your high school classes, because it can measure things that are smaller than the wavelength of light, because it uses electron beams. And in the 1950s, this allowed researchers to observe really amazing pictures, which we'll see on the next slide. So the chemical synapse is the junction where these neurotransmitters are released from one neuron and excite or inhibit the next neuron. And most of the synapses in the mammalian nervous system are chemical. So here on the top left is the electron microscope image that's been colored. You can see a synapse with the presynaptic cell with all the blue circles in it and the postsynaptic cell without any circles in it. These are surrounded, as you can see, by other cells. They don't live by themselves in a vacuum, but they're densely packed together with other cells. You can see the presynaptic membrane. You can see the presynaptic cells filled with these synaptic vesicles, which have been colored blue. And the synaptic vesicles are what are containing these neurotransmitters that then can be released into the synaptic cleft, this small space between the presynaptic and postsynaptic neuron, to influence the receptors on the postsynaptic neuron. So here's the presynaptic neuron coming in and connecting to the dendrite of the postsynaptic neuron in the most common type of synapse in the nervous system. And if you zoom in, you can see, it's also important to see in this electron micrograph image, these dark substances on the pre and postsynaptic membrane are the vesicles and receptors embedded in the membrane here, which you can see in this image in yellow. So some other things that you can see here are the microtubules by which these vesicles are brought to the presynaptic terminal, mitochondria, which can provide energy even out here in the presynaptic terminals. Here's the vesicles we talked about, which can sometimes be stored in larger granules, and the postsynaptic receptors, which we'll talk about extensively today. And the synaptic cleft we've already mentioned is this space between the pre and postsynaptic neurons. So presynaptic membrane is where the action potential terminates, coming down the axon where it then can release these chemical messages. Postsynaptic membrane is on the dendritic spine, which receives the chemical messages. And these can, we learned last week, either be excitatory or inhibitory postsynaptic potentials caused by a change in the receptors when they're activated by these neurotransmitters. And the synaptic cleft is the space between the two neurons, where filled with fluid where the chemical can travel between the two. We talked about synaptic vesicles and granules already, and about the postsynaptic receptors. There is another kind of receptor, or synapse, sorry, in the nervous system called a gap junction, or an electrical synapse, where instead of chemical messages being sent from one neuron to another, in this case, there is a continuous cavity of fluid between one neuron and the other, And electrical messages like EPSPs and IPSPs can therefore transmit between the neurons. These are fused together with these gap junction channels and allow an action potential to even pass from one neuron to another. Importantly, these are very fast. Whereas chemical messages, which can take a couple milliseconds to transmit from one neuron to another, can be more flexible in that they can amplify and diminish signals in these excitatory and inhibitory balancing acts. So next we'll talk about the process of neurotransmission. As the signal arrives at we'll talk about the 5 steps here. Sorry about that. We'll talk about the 5 steps of neurotransmission that are required to send a message from one neuron to the other. So first, neurotransmitters need to be synthesized either in the cell nucleus or out in the terminal here. And these require precursor chemicals either inside or outside the cell from the food that we eat. And these synthesized neurotransmitters are then packaged into vesicles. And the vesicles are then ready for release. So once action potential arrives at the end of the axon terminal, these vesicles can bind to the membrane and release their neurotransmitters via exocytosis, this binding of a vesicle to the cell membrane. So the neurotransmitter then crosses the fluid-filled synaptic cleft and binds to the postsynaptic receptor and activates or deactivates that neuron or starts a second messenger cascade, which we'll learn about in a couple minutes. And finally, the inactivation of that neurotransmitter is required. Either it's taken back up into the chemicals, taken back up into the presynaptic neuron, or it gets diffused into the extracellular space where it can be used to make other neurotransmitters in other cells. So we can think about this, what we just talked about, these 5 steps. So it has to be synthesized somewhere in the neuron, packaged and stored within vesicles in the terminal, transported to the presynaptic membrane and released when there's an action potential into the cleft. The neurotransmitter has to bind and activate receptors on the postsynaptic receptor, and then has postsynaptic membrane, then it has to be degraded and removed from the synaptic cleft so that it won't continue to activate the receptors. So these neurotransmitters, when they're synthesized, are either synthesized in the axon terminals or in the cell body. The transporters in the cell membrane pump the building blocks that we eat from our food into the cell to make these protein molecules, to make these neurotransmitters. So these protein molecule transporters actually allow the food to get pumped in and out to make the piece, to bring in the pieces that are required. And these sometimes are also synthesized out in the cell body from DNA and then transported on microtubules out to the axon terminal. The next thing is this release of neurotransmitter. So once the action potential arrives at the terminal of the axon, it opens up voltage sensitive calcium channels. So the calcium channels are only opened once the voltage reaches a certain level, and this voltage arrives via the action potential and opens up these channels. The calcium is plentiful on the extracellular space compared to the intracellular space. So the calcium enters this terminal and begins a process that starts the release of neurotransmitter. So the calcium binds to a protein called calmodulin, and it forms a complex. And this complex causes vesicles to empty their contents into the synapse and others to get ready and dock to the synapse, to the membrane. Okay, so the action potential reaches the voltage sensitive terminal where the calcium channels open up. Calcium enters the cell and binds to these proteins. They create this complex which can bind to vesicles and allow the vesicles to dock to the presynaptic membrane and also to empty their contents via exocytosis. So action potential, when it arrives, releases neurotransmitters via this mechanism. We should say now that studying these pathways in this intricate detail has allowed researchers to develop and understand drugs that can target the nervous system in interesting ways. So when the neurotransmitter is released into the synaptic cleft, it has to activate the receptors. It diffuses through the fluid-filled synaptic cleft and until it gets to the postsynaptic membrane. And these transmitter activated receptors, which we've learned a little bit about, are proteins embedded in the membrane that has binding site, but in this case, the binding site is for one of these neurotransmitters and then allows the proteins to change their shape. And depending on the particular type of receptor, a given neurotransmitter might depolarize the postsynaptic membrane, creating an excitatory action or an EPSP. It might hyperpolarize the postsynaptic membrane, causing inhibitory IPSPs, or it could create chemical reactions that modulate excitatory and inhibitory effects, or even influence other functions like the number of receptors in the membrane. It all depends for a given neurotransmitter, what kind of effect it's going to have. It depends on the nature of the receptor that it binds to. So these neurotransmitters can also bind to presynaptic receptors. We call these autoreceptors. So the neuron can detect when it's releasing neurotransmitter. So this is a presynaptic membrane receptor that responds to the transmitter that the neuron releases, an autoreceptor. Finally, the neurotransmitter can be deactivated in a number of ways. It can diffuse through the synaptic cleft into the extracellular space. It can degrade via enzymes in the extracellular space and synaptic cleft. It can via reuptake be taken back into the presynaptic neuron. And even the degraded parts of the neurotransmitter that the enzymes may have created could be reuptaken into the presynaptic neuron. And finally, astrocytes in the nearby area, which are known to create this intricate link with the synapse by holding it tightly together and creating this cavity. They can suck up the neurotransmitters as well and export them when needed to the axons. We've talked a little bit about the types of synapses present in the nervous system, but we'll see in the next slide that there's a large variety beyond just axon from one neuron connecting to the dendrites of another. And depending on the location and the type, these can have different effects. This allows the synapse to be a very versatile communication device because it can communicate different messages to different places of the neuron at different times. So by connecting differentially to the dendrites or the cell body or the axon, the transmitter can have differential effects. So here's a big list of different types of connections where neurons can release neurotransmitter. They can connect between one dendrite and the other. This is the most common one that we've talked about already. An axon from one can connect to the dendrites of another. It can release neurotransmitters right into the extracellular space. They can release neurotransmitter onto the cell body itself, or even onto another neuron that itself is connected to the cell body, an axosynaptic connection. They could connect to the axon, leaving the cell body, an axo-axonic connection. And finally, they could also even connect to the capillaries of the blood system. So they could release neurotransmitter right into the blood. And these neurotransmitter synapses can either be excitatory or inhibitory, and therefore have different effects on the cell. Excitatory synapses tend to be predominantly located up on the dendrites, and they have some morphological differences, like the roundness of their vesicles and how dense the material on their membrane is and how wide the synaptic cleft is. We'll see that the inhibitory synapses are counter to the excitatory, located more on the cell body, and can therefore act like a veto to stop any signal that might be coming in from the dendrites, and they have different morphology. Here's a better diagram showing that. The excitatory synapses are predominantly up here in the dendrites, where the inhibitory are at the cell body, and showing these morphological differences between excitatory and inhibitory, allowing you to intuit that you could probably tell with a good microscope, electron microscope, what types of connections these are. Okay, next we're going to talk about the different varieties of neurotransmitters that are present, and the different receptors that they have. This is a big laundry list of all the different chemicals in our brain and the receptors that they connect to. That is obviously much larger and more complicated than the nature of this class, but you're going to use this set of knowledge you'll learn here as a general overview of the different types of neurotransmitters. Luckily, these are all open book exams, so when we get to a big chart of neurotransmitters, you won't have to ask me if you'll have to remember them all. So there's about 50 different kinds of neurotransmitters that have been identified. Some inhibitory, can be even inhibitory in one area and excitatory in another. And one synapse may release more than one type of neurotransmitter. And it's not thought that there's a one-to-one relationship between a neurotransmitter, say like dopamine, and a particular behavior because of these complicated interactions in the nervous system. How do we know when something is a neurotransmitter? Well, it has to be synthesized in a neuron or be present in a neuron. Whenever the neuron's activated, the chemical must be released to produce a response in some target somewhere. And the same response has to be obtained if you experimentally place that chemical on the target. And you have to find a mechanism by which that chemical can be removed when its work is done, so that it doesn't continue to activate that target. And we'll see all these chemicals coming up are all fit this description. So synthesized or present in a neuron, it must be released when a response from the cell occurs. It has to create the same action that a chemical that experimentally placed on the receptor would have. And there must be some mechanism for it to be removed once its work is complete. So one good example of a simple circuit in the nervous system that uses excitatory and inhibitory neurotransmitter connections is present in the spinal cord of mammals, called a Renshaw loop. The motor neuron projects its main axon down to a muscle, where it releases excitatory acetylcholine, which activates the muscle and compresses it. And it has a second axon collateral, which releases acetylcholine onto this inhibitory neuron called a Renshaw interneuron. This Renshaw interneuron circularly loops right back to the motor neuron itself and releases a different it releases a different neurotransmitter called glycine, which is inhibitory, and at the receptors here creates inhibitory postsynaptic potentials that then makes this axon less likely to fire in the future. So this is a feedback loop that stops the motor neuron from firing too much by having this clamp that can stop it from overfiring whenever it gets too activated. because it sends the same signal that it's sending to your muscles, it sends to this interneuron that then deactivates it. Okay, so a neurotransmitter can also communicate in the reverse direction from the postsynaptic to the presynaptic membrane. And this is called retrograde messaging. And this can therefore influence the subsequent release of neurotransmitters or the reuptake of neurotransmitters. We'll see that the brain's endogenous cannabinoid system uses this kind of retrograde messaging to communicate from the postsynaptic to the presynaptic cell. There's generally thought of to be 4 classes of neurotransmitters that we can lump them together into. We'll talk about them in turn: small molecule transmitters, peptides, lipids, and gaseous transmitters. Small molecule transmitters are the most commonly talked about, and they're the quickest acting. They're synthesized in the neurons from the dietary nutrients that we eat, and then they're packaged ready in the axons. So there's food available in our neurons to make them, and they're readily made. We've heard about acetylcholine already, and you've heard about histamine. Dopamine, norepinephrine, and epinephrine are examples of amines, as well as serotonin. And there's also amino acids like glutamate and GABA, gamma-aminobutyric acid, and glycine, which we already saw in the Renshaw was inhibitory. So we've talked about these. You can see their abbreviations. 3 of these amines are synthesized in the same pathway of creation in the body. The molecule tyrosine, which we get from our diet, is converted into L-DOPA, which is then converted into dopamine, norepinephrine, and then epinephrine. So you can see this pathway influences the levels of all of these chemicals in the nervous system. And in the chapter, they talk about some treatments for dopamine disorders that use L-DOPA as this precursor to create more dopamine in the nervous system. So these enzymes, when they're synthesized, during this synthesis, there can be some rate limiting factors. So certain enzymes might be present in only a certain amount, and these can then therefore limit the amount of production of these neurotransmitters. For example, tyrosine hydroxylase is limited in this pathway. Amino acid transmitters, including glutamate, GABA, and glycine, are also considered small molecule transmitters that are created from our diet. GABA is the brain's main inhibitory transmitter, whereas glutamate is the main excitatory. And you can see that their only difference is this one carboxyl group off to the side that creates this different receptor activations of glutamate and GABA. Neuropeptides are synthesized from mRNA translation from DNA. So just like we talked about a couple of weeks ago, how proteins are created, these neuropeptides are small collections of amino acids put together into these protein links that can then act on receptors in the nervous system. So just like these other proteins, most are created in the ribosomes from the mRNA. They're packaged in the Golgi bodies into microtubules and then transported. However, because of this need to translate mRNA into the peptide, this process is much more slow and can't happen again as quickly. Here's some examples of different peptides that act on the nervous system, some of which you may have heard of. Are the endogenous opioids like dynorphin or beta-endorphin, which you get when you get a runner's high. Vasopressin and oxytocin are involved in our bonding to our children and our family. Growth hormone releasing factor is self-explanatory. Insulin also you've heard of, and somatostatin and neurokinin you might have heard of if you've gotten some blood work done. So these are different ways that the nervous system communicates using these peptides. So many of these also act as hormones that respond to stress. Some, like we saw, oxytocin, enable mothers to bond with their infants. Some regulate eating and drinking, pleasure and pain, like the opioids. Some contribute to learning. So it's thought that some of these create lasting changes in the nervous system. And opioids like morphine and heroin, we'll learn about in the chapter on drugs in a couple of months, mimic the action of these natural peptides, the natural opiates. So peptides, when they bind, don't have direct effects on the postsynaptic membrane voltage. They activate receptors that indirectly affect the cell structure and function. And you can't usually take these molecules as drugs because the digestive system degrades them. Whereas small molecule transmitters can go through the digestive system and enter the brain. Finally, lipid transmitters are considered another class, second from finally. Lipid transmitters are considered another class. One important example is endocannabinoids, which are the endogenous cannabinoid system in our brain. These are synthesized at the postsynaptic membrane, and they go retroactively back to the presynaptic membrane. So whenever a neurotransmitter is released onto the postsynaptic membrane, it sends a signal back to the presynaptic membrane to stop sending neurotransmitter, much like the Renshaw loop in the motor system. Two of the neurotransmitters identified in our nervous system are called anandamide and 2-arachidonoglycerol, and these have similar molecular structures to tetrahydrocannabidiol, THC, and CBD that are present in cannabis. And so those drugs act on these receptors. Endocannabinoids are lipophilic. They love fat, so they're not soluble in water, and therefore cannabinoids can be stored in your fat for a long time and show up in a drug test. Once they're synthesized, it diffuses across the clefts and interacts with the presynaptic membrane receptor. So as I said, this can release the amount of neurotransmitter. It can decrease the amount of neurotransmitter being released. So the CB1 receptor is the target of these cannabinoids. And importantly, both excitatory glutamate and inhibitory GABA synapses have these CB1 receptors on the presynaptic neuron. So cannabinoids act as neuromodulators, both in the excitatory and inhibitory systems. Finally, gaseous transmitters are a little different in that they're a gas. They're not stored inside vesicles. They're synthesized as needed in a cell, and they can easily cross cell membranes, like nitric oxide and carbon monoxide, and hydrogen sulfide is another one mentioned in the textbook. And so these, you can imagine, because they easily cross cell membranes, they're able to permeate across large amounts of tissue more easily. So this can create more kind of broad signaling across large regions of the brain or body. So here again are those four classes of neurotransmitters. We've just talked about small molecule neurotransmitters like dopamine, peptide transmitters, like the endorphins, the natural opiates, lipid transmitters like the endocannabinoids, and gaseous transmitters like nitric oxide. These different types of neurotransmitters have two different general types of receptors that they connect to, called ionotropic and metabotropic receptors. Ionotropic receptors are the ones we've been talking about so far. They're embedded in the membrane and they have a binding site for the neurotransmitter, and then they have a pore that can regulate the ion flowing through them. This could allow the movement of ions such as sodium or potassium or calcium across the membrane. So when a neurotransmitter attaches to the binding site, the pore opens and it changes the flow of ions. This is what we've talked about for the last couple of weeks. The neurotransmitter is floating around in the fluid here, as are these ions. So far the pore is closed, so the ions, even though they want to, they can't enter the cell. By want to, I mean there's probably a concentration and electrical gradient pushing them into the cell. When the neurotransmitter binds, the pore opens up and these cells are able to enter. These ions are able to enter the cell, hyper or depolarizing the cell in the process. Right, so they have a direct influence on the electrical change in the cell by letting in those chemicals. Opposed to these the other kind of receptor is called a metabotropic receptor. These don't open up a pore when a neurotransmitter binds to them, but they do some other things. They indirectly produce changes in nearby ion channels or in the cell. Each one of them is linked to something called a G protein that can affect other receptors. And so these G proteins travel in the cell and communicate and change the functioning of the cell. So here's two examples of these metabotropic receptors. On the left, you can see the G protein in the intracellular space here, which has three different subunits, alpha, beta, and gamma. When a neurotransmitter binds to the receptor, the G protein is activated, which can move the alpha subunit off of the G protein and have it travel through the extracellular space where it could then, for instance, bind to an ion channel and open it up. Therefore, you could see how this could create a change in the polarization of the cell and change in its propensity to have an action potential. Alternatively, maybe the cell wants to change the number of receptors in its membrane, and so it needs some sort of signal going into its nucleus to express more of these protein receptors and have them embedded in the membrane. So this alpha subunit could attach to an enzyme in the intracellular space, which might create a cascade of second messenger processes that could activate DNA or form new ion channels. So this can create lasting changes in the cell, which can take much longer by creating structural changes, whereas this can create quick electrical changes in the cellular activity. So these G proteins, as we said, have these three subunits. The alpha subunit detaches when a neurotransmitter binds to the receptor. And this alpha subunit can bind to other proteins in the cell membrane or in the cytoplasm, like the intracellular space. Second messengers are on the right of that image. These second messengers might bind to a membrane-bound channel, causing the channel, these are the kind of what types of signals the metabotropic receptors could send into the cell. The alpha subunit could bind to this channel, causing change in its structure and alter ion flow. It can initiate a reaction of intracellular processes leading to new ion channels, or it could send signals to the DNA to initiate or cease production of certain proteins. So these metabotropic receptors therefore allow for a single neurotransmitter binding to a single receptor site could create this escalating sequence of events where more and more happens just from this one connection. This amplification cascade is how many of these downstream proteins, like maybe a sodium-potassium pump in the cell membrane, become more or less plentiful in the cell. Whereas ionotropic receptors only have these local electrical influences on the cell membrane that they're embedded in. So as we've learned, not a single neurotransmitter isn't just associated with a single type of receptor. A neurotransmitter could bind to an ionotropic receptor and have excitatory effects, could have bind to a metabotropic effector and have metabotropic receptor and have inhibitory effects. Like acetylcholine, the same chemical activates ionotropic receptors on muscles, which activates them, excites them, but activates metabotropic receptors on the heart, which inhibits the functioning of the heart. And even one particular neurotransmitter that binds to these particular receptors, there might be different subtypes of that receptor. So for instance, there's many different subtypes of dopamine and serotonin receptors, these proteins that have slightly different shapes and therefore interact with dopamine and serotonin in different ways. So there might be more or less or more, or the presence of binding sites for other molecules. It might change how long the channel remains open, and it might change how it interacts with other intracellular signaling molecules. Here's a big long list of the main neurotransmitters and the ionotropic and metabotropic receptors that they bind to. Acetylcholine binds to these receptors called nicotinic acetylcholine receptors because nicotine from tobacco also binds to them. These are the receptors that are in our muscles. Dopamine, you can see, has 5 different metabotropic receptors. GABA has an ionotropic and a metabotropic receptor. Glycine activates NMDA and glycine receptors, and so on. You can see how many different types of receptors serotonin has. Next, we'll talk about the different neurotransmitter systems more generally and how they relate to behavior. We'll pick 4. We'll first talk about the peripheral nervous system and the enteric nervous system and what neurotransmitters they use, and then we'll talk about the brain and talk about 4 major neurotransmitter systems. So as we saw, a single neuron can use one neurotransmitter and a different neurotransmitter at another synapse. Different transmitters can even coexist within the same terminal. So we have to be careful to assume a simple cause and effect relationship between a neurotransmitter and a behavior. We'll see it's a much more complicated relationship. One good example of that is these acetylcholine receptors in the nervous system. The cholinergic neurons are these acetylcholine producing motor neurons, which we talked about in the Renshaw loop. Cholinergic refers to acetylcholine. So a cholinergic neuron is a neuron that uses acetylcholine as its main neurotransmitter, and we saw that these excite skeletal muscles to cause contractions in our muscles. A nicotinic acetylcholine receptor, its pores open up when acetylcholine binds to them and allow ions to flow, depolarizing the muscle fiber and making it more likely to contract. And we saw these are called nicotinic because nicotine also acts on these same receptors. So this pore, when it opens, simultaneously allows potassium to exit the cell and sodium to enter the cell. So the signal goes down, the vesicles bind, they release acetylcholine onto the synaptic cleft. Postsynaptic muscle, in this case, has receptors, these nicotinic acetylcholine receptors that the acetylcholine binds to, creating a propensity for this muscle to fire. In the parasympathetic and sympathetic divisions of our autonomic nervous system, which we recall control all of our organs like our heart and lungs and gut and spleen and liver, are differentially activated by these 2 neurotransmitter systems. We've seen that the sympathetic system rouses the body for action, increasing our heart rate and decreasing our digestive system. The parasympathetic system, when we get home from a hard day at work, calms our body down, and the digestive system ramps up and the heart rate ramps down. Both of these divisions are controlled by acetylcholine neurons that leave the central nervous system at 2 different levels of the spinal cord. So the parasympathetic neurons, sorry, these acetylcholine neurons then synapse with either parasympathetic neurons that also themselves contain acetylcholine and make you more likely to rest, or sympathetic neurons that contain norepinephrine and stimulate the target organ to make it more likely to ready to fight or flight. So here's a diagrammatic representation of that. This parasympathetic division has acetylcholine neurons that leave the central nervous system, synapse onto second neurons that make the target organ relax, like the digestive system turn on and the heart rate go down. Contrary to this, the sympathetic system has acetylcholine neurons that synapse onto these norepinephrine producing neurons, and these norepinephrine producing neurons stimulate the target organs in the various ways to increase heart rate or decrease digestion. Right, so the same norepinephrine chemical can have an excitatory effect to increase heart rate and an inhibitory effect to decrease digestion, depending on the type of receptor it binds to in the target organ. We've said this already, I think. So these CNS neurons synapse with either parasympathetic neurons that contain acetylcholine. These acetylcholine receptors, for instance, on the heart are inhibitory, making the heart slow down. In the gut, they're excitatory, making our digestion increase. The sympathetic neurons that get connected to by other central nervous system neurons contain norepinephrine, and they release them onto target organs as well. And these norepinephrine, when it reaches the heart, is excitatory, whereas norepinephrine, when it reaches the gut, is inhibitory, causing you to not digest too much when you're ready to run away. The enteric nervous system in our gut has its own autonomy. It can act without any input from the central nervous system. It uses all 4 classes of neurotransmitters we've learned about, and up to 30 different types of neurotransmitters. It uses serotonin and dopamine. And there's also gap junctions in the enteric nervous system as well. Finally, we'll talk about the central nervous system. So we've talked a little bit about the reticular activating system in the brainstem. This collection of neurons that project into the brain and create our wakeful cycles and our general consciousness of the day changes. And so these activating systems coordinate large amounts of brain activity by sending a neurotransmitter throughout the brain. The cell bodies that produce these axons that travel throughout the brain are in the brainstem, and their axons project widely. We'll learn about 4 systems: the cholinergic acetylcholine system, the dopaminergic dopamine system, the noradrenergic norepinephrine system, and the serotonergic serotonin system. The cholinergic system is involved in our normal waking behavior. And it's thought to be related to attention and memory. And loss of cholinergic neurons in the brainstem is associated with Alzheimer's disease. The dopaminergic system, or the dopamine system, has 2 pathways: the nigrostriatal and the mesolimbic. The nigrostriatal is involved in coordinating our movement with our basal ganglia and is affected in Parkinson's disease. And the mesolimbic pathway is involved in addiction and our aversion or desire for environmental stimuli. So here's a diagram showing those cholinergic and dopaminergic systems. On the top, the cholinergic system has these midbrain nuclei and basal forebrain nuclei that release acetylcholine throughout the brain. Here's the 2 dopaminergic systems: the nigrostriatal pathway from the substantia nigra in the brainstem up into the basal ganglia, and the mesolimbic pathway from the ventral tegmentum into the frontal cortex and the cerebellum. The nigrostriatal pathway seems to be involved in our normal motor behavior because the loss of dopamine is involved in motor problems of Parkinson's disease. And the dopamine system that goes into our frontal lobes is involved in our feelings of rewards and pleasure, and we'll see is related to our propensity to desire drugs of habit. And these are both been implicated. This has been implicated in schizophrenia, and they've both been related to changes in attention. The noradrenergic system, or the norepinephrine system, plays a role in learning by making neurons change their structure when they've been involved in a learning episode, and might help normal development of the brain and organizing movements. Imbalances are associated with depression and mania. Finally, the serotonergic system plays a role in wakefulness and learning. Imbalances of serotonin we know are associated with depression and schizophrenia and OCD, sleep apnea, and sudden infant death syndrome. Here's two diagrams showing the two systems. The noradrenergic system starts in a locus coeruleus in the brainstem and transmits throughout the frontal lobe cerebellum. Decreases in activity are associated with depression, and increases are associated with mania, and decreases associated also with ADHD. The serotonergic system has these areas in the brainstem called the raphe nuclei, which create axons that project serotonin throughout the brain. They're related to OCD and tics, and decreases in serotonin is related to depression. And we'll see in our chapter on drugs which different drugs activate which different ones of these systems. Finally, today we're going to talk about briefly about changes that can happen in the synapse with learning and memory, and how this can set the stage for our very adaptive and flexible brains. We'll introduce the concepts which you've probably learned about in other classes. Learning is a relatively permanent change in behavior that results from experience. And neuroplasticity is a nervous system's potential for changing, which enhances its ability to adapt. So a nervous system that can change can adapt to different situations. Neuroplasticity is required for learning and memory so that our nervous system reacts differently in the future to the same stimulus. It's been proposed for a while now that the location by which memories are created in the brain is the synapse. And Donald Hebb, a famous Canadian neuroscientist, proposed that when the axon of cell A is near enough to excite a cell B and repeatedly or persistently take part in firing it, when cells fire together, some growth process or metabolic change takes place in one or both cells, such that A's efficiency as one of the cells firing B is increased. They become wired together. So cells that fire together wire together. The idea is that when two cells are active together, their connection should be strengthened so that in the future they're more likely to activate one another. This research also went on to lead to a Nobel Prize by Eric Kandel, who studied how learning occurred in the neurons of an animal called a plesia, a sea slug, because of the giant axon size in this animal and the ease of dissecting the axon. So what he did was he looked at very simple defensive behaviors in the sea slug, and that changed over time, and looked at the nervous system of the sea slug to see where those changes were represented. We'll talk about two types of learning: habituation and sensitization. Habituation is a learning behavior in which a stimulus response weakens over repeated presentations of the stimulus. So the noise of the fan in the room where you're in right now, over time, your nervous system comes to represent that more weakly as it becomes habituated to the stimulus. In the aplesia, and a good example of this to study for Eric Kandel was the gill withdrawal response, whereas this gill that's used to pull oxygen from the water is withdrawn when something scary happens to the aplesia. So at first, when a jet of water is sprayed on the aplesia, the gill withdraws into the animal, but after repeated stimulation of that water jet, the same water jet no longer leads the gill to withdraw into the animal. So the sensory neuron stimulates the motor neuron before habituation. The shot from the water jet stimulates the motor neuron and makes it likely to fire and retract the gill. So with habituation, the EPSPs, it was measured by Eric Kandel, the amount of excitation from a given input becomes smaller and smaller. So the motor, so if we measure from this motor neuron and we stimulate the sensory neuron here with water, electricity, the depolarization of the motor neuron becomes smaller and smaller with repeated stimulation of this sensory neuron. The motor neuron is receiving less neurotransmitter from the sensory neuron across the synapse, leading to these smaller EPSPs. So the habituation must be taking place in the axon terminal of the sensory neuron so that it comes to not release as much neurotransmitter. So it was discovered that with habituation, the influx of calcium ions decreases, leading to less neurotransmitter release at the presynaptic neuron, and therefore less depolarization of the postsynaptic membrane. So habituation is created by this change in the amount of calcium entering the cell for a given action potential. So a same amount of voltage change with a given action potential causes less calcium influx. This can be due to reduced sensitivity of the calcium channels and also a decreased release of neurotransmitter in the cell. Alternatively, another type of learning that the animal can do is called sensitization, and that we can do is called sensitization. We can become more sensitive to a stimulus over time. So the response can strengthen with repeated stimulation because the stimulus at first was so novel or stronger than normal. So a single shock to the tail instead of just a little water jet can actually enhance the gill withdrawal response. So instead of repeated water jets causing the gill withdrawal to get smaller, a large shock somewhere in that process can cause the withdrawal response to be enhanced. Think about this maybe with respect to PTSD and people's increased sensory overload after suffering from a stressful situation. So originally, in response to an action potential on the sensory neuron, calcium channels are slow to open. So the interneuron was where, so the sensory neuron was what picked up the siphon of water and caused the gill to withdraw. The interneuron picks up the electrical stimulation and releases serotonin onto that siphon sensory neuron. So it's a connection directly between a neuron and the axon terminal of another. So these potassium channels are slow to open when the sensory neuron fires the motor neuron. And this slower opening causes the membrane to not repolarize as quickly, so the action potential lasts longer. And this prolongs the inflow of calcium and leads to more transmitter being released. So at the molecular level and behavioral level, they're opposites, sensitization and habituation. In sensitization, more calcium results in more transmitter being released. In habituation, less calcium results in less transmitter being released. So here in sensitization, in this model, the serotonin reduces the potassium efflux, the exit of potassium through potassium channels, prolonging an action potential in this area, leading these calcium channels to be open for longer. so that more neurotransmitter is released, causing a greater depolarization of the membrane, in this case, causing a greater gill withdrawal response. So when we study changes with learning in the nervous system, we have to consider that they need to be long enough to account for the permanent changes in behavior. So it can't just be a quick change in the firing of a cell. It has to maybe be a structural change that lasts over time. So these habituation and sensitization effects can last for months at a time. So one mechanism by which these learning effects can occur is by a change in the number and size of the synapses in habituated and sensitized aplesias. So here is a habituated and a sensitized aplesia. The sensory neuron, which normally activates this motor neuron, in a habituated animal has less axon connections, and a sensitized animal comes to grow more. And you could see how this would increasingly or decreasingly activate that motor neuron. In order to change the synapse number and size, transcription and translation of DNA has to occur to create these lasting structural changes. And so therefore, second messenger systems are going to play an important role. These metabotropic receptors that connect into cells' DNA and change the expression of certain receptors in their membranes. Learning can affect the axonal transport. It can change the number of vesicles, change the size of the cleft, change the dendrite width and length, increase the terminal size. It can make there be more dense contact areas in the cell. It can increase the size of the spines, and it can even increase transport for spine construction so that more spines are created in the dendrites. That brings us to the end of chapter 5 and everything that we're going to cover for the first midterm. Good luck studying and reading the book.